Welcome, my dear students. Today we begin exploring the very foundation of every protein, the primary structure. Just like a long sentence is built from letters arranged in a specific order, a protein begins as a precise sequence of amino acids linked one after another. This unique order decides everything the protein will eventually become: its shape, its function, and even its stability. In this class, we will decode how this simple linear chain holds the blueprint for life's most complex biological machines. So let's uncover how the primary structure sets the stage for all higher levels of protein organization. So the primary structure or primary protein structure is the sequence of a chain of amino acids. The primary structure is the exact linear order of amino acids in a polypeptide chain. We can ask, what is the primary structure of protein? Amino acids are linked one by peptide bonds, forming a long chain with a unique with a specific sequence. This sequence is the foundation of all higher structural levels such as secondary, tertiary, and quaternary, and it ultimately determines the protein shape, stability, and function. How the primary structure is formed? Number one is the peptide bond formation. We know that a peptide bond is a strong covalent bond formed when the carboxyl group bond COOH of one amino acid reacts with the amino group or bond NH2 of another amino acid. This reaction releases one molecule of water, which specifies dehydration and condensation reaction, and produces a growing polypeptide chain. Directionality of proteins always have two fixed ends, that is N terminus or amino terminus, which has a fixed or free amino group, and C terminus, which is the carboxy terminus, has a free bond COOH group. The sequence is always written from N to C direction, just as the ribosome synthesizes the protein molecule. Key features of the primary structure: unique sequence for every protein. Primary structure determines, or it gives every protein has a specific sequence. Even a single amino acid change can alter its function dramatically. Example: in sickle cell anemia, glutamic acid at position 6 in the beta chain of hemoglobin is replaced by valine. Variable length: proteins differ widely in size. Short proteins, only a few amino acids. Medium length, that is ribonuclease, that is 124 amino acids long. Very large protein is the titin, that is approximately 34,000 amino acid long, which is the largest known protein. Primary structure also determines protein folding and function of protein. The sequence determines how the chain folds into a alpha helices and beta sheets, that which are the secondary structure. It determines the 3D shape or the tertiary structure of a protein. It is responsible for interaction with other proteins such as the quaternary structure. So golden rule is the sequence determine the protein structure, and protein structure determine the ultimate protein function. Stability of a protein: primary structure is held by strong peptide bonds, making it the most stable level. Heat, acids, or chemicals may denature protein shape but do not break peptide bonds unless conditions are extreme. How scientists determine primary structure? First of all is the Edman degradation method that removes one amino acid at a time from the N terminus. It used mainly for short peptides. Beside Edman degradation, there is a process known as mass spectrometry, which is a highly accurate and fast process. It identifies amino acids based on mass to charge ratio, and it is the most widely used technique. DNA sequencing: since DNA encodes protein sequence, amino acid sequence, so the decoding the gene or decoding the nucleotide sequence of a DNA reveals the exact amino acid order within a protein. Biological significance of primary structure: first of all is the protein folding. Correct sequences of amino acid ensure correct shape, which is vital for enzymes, hormones, or transporter. Functional specificity: the sequence determine the active site of enzymes, antigen binding sites in antibodies, etc. Mutations and disease: a change in DNA altered sequence, defective protein, and examples such as sickle cell anemia, cystic fibrosis, etc. Evolutionary clue: comparing sequences helps study in evolutionary relationship among species. Protein engineering and biotechnology: it is responsible for modifying sequences, help design better enzymes, therapeutic proteins, and industrial molecules. Important examples of the primary structure of protein: first of all is the insulin, which has a 2 polypeptide chain. A chain has 21 amino acids and B chain has 30 amino acids. The specific arrangement is crucial for glucose regulation. Sickle cell hemoglobin: there is a single mutation, glutamic acid to valine at position 6 of the beta chain. It causes hemoglobin to polymerize. It causes sickle cell RBCs and ultimately it causes the disease. So for quick revision purpose, we can say that protein structure is equal to the sequence of amino acids within a protein. It is linked by peptide bonds. The direction of amino acids within a protein is N-terminus to C-terminus, and the sequence always determines the protein folding and the function of the protein. The primary structure is highly stable due to its covalent bonds, and it can be identified by Edman degradation, mass spectrometry, and DNA sequencing processes. Single amino acid change may cause major disorders within a primary structure of protein. So as we conclude, remember that the primary structure may look simple, but it carries extraordinary power. By determining which amino acid comes next in the chain, the sequence dictates how the protein will fold, how it will function, and how it will interact inside the cell. Even a single change in the sequence can transform the entire protein's structure and protein's behavior. So the primary structure is not just the beginning, it is the master code that shapes the identity of every protein. Keep this foundation strong and the rest of the protein architecture becomes beautifully clear. Thank you.